Jake McGreig, University of Kent. Hi everyone, welcome to my presentation looking at differentially conserved amino acid positions and how they reflect differences in SARS-CoV-2 and SARS-CoV behaviour. All of the work I'm going to speak to you about today is available in bioRxiv with the following title. And I'll be showing this slide at the end of the presentation so you can look into it in more detail if you want to. So what is SARS-CoV-2? So SARS-CoV-2 is responsible for the global pandemic at the moment, with over 9.4 million cases worldwide and over 481,000 deaths worldwide as of the 25th of June 2020. So coronavirus is part of the Coronaviridae family and is a positive sense single-stranded RNA virus, which is around 29,000 base pairs long and made up of 29 proteins. And 23 of these proteins share homology with SARS-CoV, an outbreak that occurred in 2002 and 2004, which is also part of the Coronaviridae family, but it had a very different phenotype. So the proteins which make up SARS-CoV-2, of the 29 of them, there are 16 non-structural proteins, some accessory proteins, and some structural proteins which are shown in the figure on the right-hand side. So the envelope protein, the spike protein, which is involved in mediating virus cell entry by binding to the ACE2 receptor, the nucleic acid protein, and the membrane protein. So SARS-CoV and SARS-CoV-2, despite being of the same virus family and sharing homology in a large proportion of their proteins, they actually have very different phenotypes. So SARS-CoV infects the lower respiratory tract, has a mortality rate of around 1 in 10, and has a low transmission rate, whereas SARS-CoV-2 invades the upper respiratory tract, has a low mortality rate, but a very high transmission rate, which is how it's able to spread so easily across the globe. So what we wanted to do was compare these two proteins to try and identify sequence positions in the proteins which could be responsible for the phenotypic differences. And to do this, we used differentially conserved positions. But what are differentially conserved positions? So these are positions between two groups, so you can see here group 1 and group 2, that are conserved, so we can see all V's and all K at position 3, so they're both conserved, and there is no overlap between group 1 and group 2. So that's how we're terming DCPs. So this method has been applied previously in work published shown at the sites below, and in this work we looked at applying it to Ebola virus. So there are several pathogenic species in Ebola virus and one non-pathogenic species. And so we compared them and identified DCPs between them to try and work out where that source of pathogenicity was coming from. So, with this being a valid method to use to identify these sorts of protein sequence differences, we then applied it to coronavirus. So first we obtained the SARS-CoV-2 sequences from Gizzade and the SARS-CoV sequences from NCBI. So we translated the nucleotide sequences we got from Gizzade into proteins by using getorf and extracting the proteins from the open reading frames. We then pre-processed the sequences, so this removed poor quality sequences, so things with lots of X's or gaps. We then aligned them using Muscle, which is alignment software. Then we calculated the consensus residues and frequencies at each position in the alignment. We then identified the conserved positions in SARS-CoV and SARS-CoV-2, and we used this information to then label the DCPs. And here's what we found. So we saw a large proportion of DCPs in the spike protein and non-structural protein 3, which is unsurprising as they are the largest of all the proteins in coronavirus. But we also saw that there was an enrichment in these proteins as well of DCPs, so it covers 18.54% and 15.73% for each of these respectively. So in particular, we were interested in the spike protein because it's been shown to be key for facilitating virus entry into the host. So we wanted to identify if any positions in the spike protein were in functional sites or sites of interest. So we had a look at where the DCPs were found in the spike protein. So in this figure, in red, we have the DCPs which span across the spike protein. In grey, we have DCPs that we identified between the two reference sequences for SARS-CoV and SARS-CoV-2, but were then no longer classed as DCPs when the alignments are fully populated. So it just shows that there is variation that occurs in the SARS-CoV-2 sequences. Then we have this inset of the receptor binding domain of the spike protein, so you can see in a bit more detail, where there are DCPs spanning the whole receptor binding domain, but there seems to be quite a lot located between 45 and 500. And finally, this inset here shows the position, the position with a square around it, so these arginines. So these are residues which bind to serine protease TMP RSS2, and this is vital for the activation of the spike protein. So it's a protease cleavage site, and we'll come on to that later. So we wanted to look at the DCPs and the interface between ACE2, so the receptor that the spike protein binds to, and the spike protein, to see whether there were lots of changes there. So we have the ACE2 in cyan and spike protein in grey. And any DCP that's within 5 angstroms of ACE2, we've then highlighted in red. And in bold, we can see residues, so T501N and N493Q, which have been shown to be pivotal to the interaction between ACE2 and the spike protein in other research. So these differences at the receptor binding interface could be responsible for the altered transmission of SARS-CoV and SARS-CoV-2. In addition, position R426 in SARS-CoV is able to form a hydrogen bond with Q325 in ACE2, whereas in SARS-CoV-2, N439, which is the equivalent position in the alignment, is unable to form a hydrogen bond with Q325 as the distance is too great. So this could also indicate that transmission is affected as the binding interaction would be different. Lastly, we look at the drug sensitivity profiles of SARS-CoV and SARS-CoV-2, and this work was performed by our collaborators in Frankfurt. A colorectal cancer cell line was used in this work and infected with SARS and SARS-CoV-2. Several drugs were tested, and today we're going to be looking at the effect of the serine protease inhibitors and what effect they have on the two viruses, as we identify DCPs near the serine protease cleavage site. These drugs are all approved and have various applications today. Camostat and nifamostat are used to treat pancreatitis, and aprotinin is used to prevent blood loss in surgery and is also used to treat influenza, so they all show some antivirus activity. So as you can see in the graphs, which show the cytoprotective effect inhibition on the viruses, this shows that SARS-2 is more sensitive to serine protease than SARS. So this shows that wherever you see the SARS-CoV-2 in red above SARS-CoV in black, it shows that SARS-CoV-2 is more sensitive to these drugs than SARS-CoV is. In addition, when aprotinin, so the one at the bottom, is combined with omeprazole, you can see an even greater sensitivity for SARS-CoV-2. So in addition to this, the cells were also stained for double-stranded RNA, which shows the presence of the virus, and these were compared to a virus control. So the staining in red shows the presence of the virus. So as you can see, the amount of staining in camostat and nifamostat goes up, so you can see that it looks like there's more virus here, but this is most likely due to the cells aren't dying, so therefore more virus is stained, so you can see more virus. Whereas in aprotinin, you see a great reduction in staining, so less presence of virus. So this means that aprotinin could be a promising drug candidate moving forward. as it shows that the SARS-CoV-2 seems quite sensitive to it. So to conclude, we identified around 119 out of 783 DCPs that we predict to have a functional effect in between SARS and SARS-CoV-2. We saw an enrichment of DCPs in 3A, P6, the non-structural proteins 2, 3, and 4, and the spike protein, where DCPs, or differentially conserved positions, in the spike protein receptor binding domain may be responsible for the increased transmission of SARS-CoV-2. However, other DCPs may also be the cause of the SARS-CoV-2 phenotype, where it's got a much higher transmission. But the virus behaviour may also be due to immune response inhibition or other host factors. And lastly, there are some accessory proteins which aren't considered in this study, as they lacked a shared homology with SARS-CoV, but we do cover the vast majority of the proteins, so 23 out of 29 of them, and most of these, we feel like this is enough to get the vast majority of the virus protein functions compared. So yeah, thank you for listening. I'd like to thank everyone in my lab group, everyone who worked in this project, so Martin Michaelis, Mark West, Stuart Marston, and Katie May McLaughlin, and all of our collaborators in Frankfurt. And if you want more information on the paper, it's available with the following title in DOI as shown on this slide. And thank you, and I welcome any questions.